Thank you, Frank. Yes, indeed, my name is Katrien. Happy to be here. Happy to be the very last one of the of the session. And I'm pleased to introduce you our company, PenCancer-T. We develop T-cell therapies indeed, and we build actually forward on the research that was developed by Erasmus MC, the biggest university hospital in the Netherlands. Today we heard a lot about immuno-oncology, so I'm at the right spots. And yes, T-cell therapy really is a game changer in the fields. It's a credible and validated modality that has induced really deep and long-lived remissions, often with one single injection. And so it relies actually on the gene engineering and redirection of the T cells, which are actually the main immune suppressor cells, against tumors. The majority today, as you may know, were actually accumulated by CAR-T companies against liquid tumors. And it is really incredible. I mean, some patients reported to be still in remission 10 years after they were diagnosed with leukemia and treated with a CAR-T. So it is unprecedented. Unfortunately, CAR-T is not so successful in solid cancer. And that is where TCR comes in. Because TCR therapies have demonstrated their clinical proof of concept in multiple solid cancers, albeit rare solid cancers and in a limited number of targets. So if we really want to progress this huge potential of this kind of therapies, then we need to overcome like two main obstacles in our view, which are expanding the number of therapeutic targets. For us, we believe that the targets for T cells are the prerequisite of a safe and effective treatment. And two, we need to increase the duration of the T cell responses. As my peers in the previous speech alluded to, this microenvironment actually has a suppressive role and jeopardizes the T cells from optimally functioning. So we need to help these T cells to function better and to, have their real potential unleashed. And that's exactly what we do at PenCancer-T. We actually address these two major obstacles into one product. Hence, dual acting approach. Our T cells target novel tumor targets and at the same time are engineered to enhance the durability of their response. So PenCancer-T has an discovery engine, and has discovered 30 novel proprietary targets that are selectively and robustly expressed across 10 major solid tumors. One example is the lead target PCT1, which is shared by 90% of patients with triple-negative breast cancer. This is really a devastating, aggressive form of breast cancer affecting young women. It's very heterogeneous and it lacks good targets. That's why the treatment options so far are very limited up to now. So, secondly, we aim to make our therapies long-lived and to overcome that immune suppression. And our approach is actually to understand, firstly, the immune profile of a given tumor. This is very different from tumor to tumor. So our platform actually integrates advanced phenotyping and allows us to characterize the most dominant suppressing mechanism for the tumors of interest. And that information, that intelligence then actually guides our engineering strategy with the idea to make the T cell resistant to the immune suppression. So there's other companies who are making products to modulate the tumor microenvironments. That's not what we do. We want to make our T cell resisting to these hostile influences. And so we're developing actually multiple solutions depending on the immune profile of the tumors. The one that is most mature, let's say, is one where we introduced a co-stimulatory domain into the T-cell receptor. And this has showed actually already enhanced persistence and durability, and is now being tested in triple-negative breast cancer model. Putting it all together, we have 3 platforms that actually together forms our products, which is one target discovery platform where we look for immunogene targets, in fact, safe targets as well, and TCR discovery, yielding TCRs that are highly specific, non-cross-reactive, and that binds with an optimal affinity to our targets. And this dual-acting engineering platform, making our T cells resistant to the suppression. So, putting it all together, our lead program exemplifies actually this dual approach. We have demonstrated a proof of concept, a preclinical proof of concept, and selected our clinical TCR lead, which is a highly specific and effective TCR that binds that novel target, which we call PCT1. So this product is, has compelling data and can be moved to the clinic. While in parallel, we're actually pushing forward that co-stimulatory version of the TCR, and that's now being tested in a triple-negative breast cancer model. We're ambitious, so this is the beginning. In the end, we want to then move to other solid tumors, but first and work on triple-negative breast cancer. We're scientists, so just showing, seeing is still believing, some proof of concept data. So here you see an in vitro 3-dimensional triple-negative breast cancer tumoroid in green. And so on the left side, you see what happens if we co-culture this tumoroid with mock T cells, which are unengineered. And you see, in fact, after 48 hours that the tumor grows and almost explodes. It's almost like a Picasso, in fact. Whereas on the right side, you see what happens when our engineered T cells are co-cultured. And you see that the tumoroid totally regresses. So, and the cell death signal appears. So this is very promising. Another example, this is in an immune incompetent mice model where we grew triple-negative breast cancer tumors. And at the apex, we injected the T cells. So in gray, you see what happens if the mock T cells are given, the tumor just continues to grow. Whereas in blue, after injecting our T cells, in fact, the tumor regresses nicely. Visualizing all our plans. So today we have a lead program with a selected clinical TCR candidate, which is highly specific and effective, binding against the novel target PCT1. In parallel, we are also testing this co-stimulated version of the TCR. And then in the coming 5 years, we are also planning to exploit other targets, other cancers, and other immune approaches. I'm not alone. I'm backed by a strong, passionate team. So I collaborate with my colleagues, Professor Reno de Betz, who is Professor Immunology at Erasmus, founder and CSO, Dr. Tom Holdig, our medic, and he has a serious track record in T-cell drug development, worked at Adaptimmune. Dr. Dorel Hamrel, she's the co-founder and oversees the R&D activities. Reinoud Hesselink, who is translating the science into a product. And then David, our financial guy. So together with our supervisory board and our scientific experts, we're building this company, and we couldn't do this, of course, without our investors. So we're backed by Van Herk, Tuya, Swambridge, and Erasmus. And so, to conclude, pan-cancer T is in a unique position by the combination of these novel tumor targets. Yes, the technology to overcome this immune suppression. We have a preclinical proof of concept and selected the clinical TCR candidates. And I have the luxury to work with a splendid, experienced team. And we are all determined to make game-changing T-cell therapy.